Here's the latest audio research news from the transmitter. New dopamine sensor powers 3-color imaging in live animals. By Diana Kwan. A new sensor makes it possible for the first time to simultaneously track dopamine and up to 2 additional molecules in the brains of living animals. The sensor, dubbed Halo DA1, uses a novel dopamine tagging system that emits light at the far red end of the color spectrum, according to the team behind the work. There's a real need to monitor multiple relevant molecules, as they're doing here, says Nicholas Trich, assistant professor of neuroscience at McGill University, who was not involved in the study. Because dopamine is involved in a range of key brain functions, when studying its effects on a cell, it's important to consider other neuromodulators that are released at the same time, as well as the signaling cascades these molecules may trigger, Trich says. Most dopamine tracking strategies genetically encode a naturally occurring fluorescent protein into dopamine receptors. When dopamine attaches to the modified receptors, the fluorescent protein changes shape and emits light. But naturally occurring fluorescent proteins have a limited color palette, which has made it difficult to develop sensors that can go beyond 2-color imaging, says study investigator Yulong Li, professor of life sciences at Peking University. Instead of genetically encoding a fluorescent protein, Halo DA1 attaches a synthetic molecule called halotag to dopamine receptors. This tag binds tightly to previously developed artificial dyes that change shape and fluoresce in the far red spectrum when dopamine binds to its receptors. Because the dyes fluoresce at the far end of the red spectrum, it leaves room for other sensors to glow at different wavelengths. Other scientists have previously combined halotag and synthetic dyes to create sensors, but this is the first time halotag has been applied to a member of the family of G-protein coupled receptors GPCRs, namely dopamine receptors. It opens up so many new possibilities because there are so many GPCRs that play critical roles in brain function, says Robert Campbell, professor of chemistry at the University of Tokyo, who was not involved in this study. Lee's team tested the sensor in living zebrafish and mice. The researchers injected a plasmid carrying the sensor into fish larvae and then submerged the fish in the dye. For the mice, they injected a virus encoding the Halo DA1 into select areas of the brain and then injected the dye, which can cross the blood-brain barrier, into the bloodstream. In a variety of tests, the team was able to use Halo DA1 together with existing sensors to generate 3-color imaging of dopamine and 2 other molecules, including acetylcholine, calcium, and adenosine triphosphate ATP. In mice, the researchers combined the sensor with optogenetics to monitor dopamine and calcium release in response to the stimulation of specific brain regions. They also used the tool to simultaneously track dopamine, acetylcholine, and the secondary messenger cyclic adenosine monophosphate camp in the nucleus accumbens in response to the mice consuming sugar or experiencing mild foot shocks. The experiments revealed how these positive and negative stimuli change dopamine and acetylcholine levels, and how that, in turn, can affect the production of camp. The researchers published their findings last month in Science. A major technological advance in this study was the discovery of a dye that, compared with other dyes researchers have tested, can more readily reach the brain, Campbell says. Other groups, including his own, can now apply this dye to their own sensors, he adds. Halo DA1 has some limitations, Lee notes. One is that, unlike genetically encoded fluorescent proteins, which are long-lasting, the dye used in this sensor fades after a few days. Researchers who want to carry out longer experiments may need to re-inject the dye periodically. Lee says his group is working on finding non-invasive ways to sustainably provide enough dye for these lengthier projects. Another limitation is that not all confocal microscopes, which are commonly used in these studies, are set up to conduct 3-color imaging, Trich says. At the same time, though, the ability to use synthetic dyes mean that we're not limited to things that our bodies can engineer, he adds. This is an exciting new development that I'm sure in a few years from now we'll look back at and think how it really opened a whole new area of imaging. This audio was created using artificial intelligence. Stay up to date with the latest neuroscience news and perspectives at thetransmitter.org. Follow audio research news on Spotify, YouTube, or wherever you get your podcasts.